The lipid bilayer's amphipathic nature gives rise to its inability to unite with either the extracellular or intracellular fluid. Therefore, it provides a barrier for the movement of substances between the extracellular and intracellular compartments. The pathways by which the substances can cross the membrane include passive diffusion, facilitated diffusion, primary active transport, and secondary active transport. Passive diffusion is the net movement of material from an area of high concentration to an area of lower concentration. The difference in concentration between the two areas is known as the concentration gradient. Diffusion will continue until the gradient has been eliminated. This pathway does not require ATP or a carrier protein and is taken by substances that are lipid soluble. Examples include oxygen, nitrogen, and carbon dioxide. These substances are delivered to the interior of the cell as if the cell membrane did not exist. Ions, on the other hand, diffuse across the membrane via the integral proteins that form channels. These channels conduct a specific species of ion, such as sodium or potassium. They can differentiate between size and charge of the molecule being diffused. In some ion channels, passage through the pore is governed by a gate, which may be opened or closed by chemical or electrical signals or mechanical force, depending on the type of channel. In a process known as facilitated diffusion, a special carrier protein with a central channel acts to help molecules move across the membrane. These special carrier molecules that form the protein channel bind only to a specific molecule, such as a particular sugar or amino acid. Once the molecule binds to the carrier protein, this protein helps or facilitates the diffusion process by changing its shape to move the molecule down its concentration gradient through the membrane, releasing it inside the cell. Active transport occurs when a cell membrane moves molecules or ions against its concentration gradient. This mechanism has two types and is classified into primary or secondary active transport. The primary active transport mechanism that has been studied in the greatest detail is a sodium-potassium pump. In this pump, 3 sodium ions bind on the inside of the carrier protein, causing the ATPase function of the protein to become activated. The ATPase then cleaves one molecule of ATP and splits it into adenosine diphosphate and high-energy phosphate. This energy is believed to cause a conformational change in the protein carrier molecule, releasing the sodium ions to the outside of the cell and the potassium ions to the inside of the cell. Through primary active transport, a large concentration gradient of sodium results due to a high concentration of sodium outside and a low concentration of sodium inside the cell. This electrochemical gradient represents a storehouse of energy due to the excess sodium outside the cell membrane, which is always attempting to diffuse into the cell. The diffusion energy of sodium can pull other substances along with sodium through the cell membrane. This phenomenon is called cotransport or symport. This is a form of secondary active transport. For sodium to pull another substance along with it, a coupling mechanism is required. This is achieved by means of another carrier protein in the cell membrane. The carrier serves as an attachment point for both the sodium ion and the substance to be cotransported. Once they are both attached, a conformational change occurs in the carrier protein, and the energy gradient of the sodium ion causes both the sodium ion and the other substance to be transported together to the interior of the cell. In countertransport or antiport, sodium ions again attempt to diffuse into the interior of the cell because of their large concentration gradient. The other substance is located inside the cell and must be moved to the outside. The sodium ions bind to the exterior portion of the carrier protein, while the countertransported substance binds to the inferior portion of the carrier protein. Once both have bound, a conformational change occurs with the energy of the sodium ion moving to the inside, causing the other substance to move to the outside of the cell. Glucose is transported from the intestinal lumen into the cells by a sodium-dependent co-transport in the luminal membrane. Active transport of sodium through the basolateral membrane depletes the sodium inside the cells. This low concentration of sodium inside the cells maintains the concentration gradient. It causes the sodium ions in the intestinal lumen to combine with the transport protein. However, the transport protein will not bring the sodium to the interior of the cell until it combines with another appropriate substance such as glucose. Both the sodium and glucose are transported inside the cell. This physiologic principle explains the role of glucose in oral rehydrating solutions. Glucose is a co-transport of electrolytes in the body. Sugar is important to improve absorption of sodium and water. However, if there is too much sugar present in solution, diarrhea and dehydration can worsen due to an osmotic diuretic component. The transport pathway mechanisms can be summarized as follows. In passive diffusion through the lipid bilayer, substances move down the concentration gradient, do not require ATP, cannot be saturated, and is not chemical specific. In passive diffusion using channels, substances also move down the concentration gradient and do not require ATP. However, it can be saturated and is chemical specific. In facilitated diffusion, substances move down the concentration gradient and do not require ATP. However, they do require a carrier protein. It can be saturated and is chemical specific. In primary active transport, substances move against a concentration gradient and require energy in the form of ATP. It can be saturated and is also chemical specific. In secondary active transport, substances move against the concentration gradient and require energy from the movement of one substance. Co-transport or symport entails solutes moving in the same direction, while counter-transport or antiport entails solutes moving in the opposite directions.